in cerebral hemodynamics after the effect of probiotics. We are doing a study on Parkinson's patients also, and to evaluate the microbiome, and as has been studied, the geographical indicators are also important. Microbiome of an Indian Parkinson's may be very different from the microbiome of a Parkinson's abroad, and these are the things to be taken into consideration. Recently, a very good paper in Cell did establish that signals from gut microbes are required for neuroinflammatory responses, as well as they hallmark the GI and motor deficits, both GI deficits as well as movement related deficits, which are seen in a model of Parkinson's disease, where a whole lot of nice things were done, which is a model of an elegant study design. That is, the germ-free animal model was then implanted with microbiome from Parkinson's patients and seen, and everything was seen in the GIT, in the brain. So gut microbes promote the alpha-synuclein mediated motor deficits and brain pathology, and the gut symptoms arise much earlier than the motor symptoms which manifest. Depletion of these gut bacteria would reduce the normal microglia activation. The small chain fatty acids would modulate these microglia and enhance the pathophysiology, and human gut microbiota from the Parkinson's patients induce enhanced motor dysfunction in mice. So there is a good link between the enteric nervous system, the gut dysbiosis, the intestinal permeability changes. Lot of good papers, including in Nature and Cell, have come up in the past 5 years on this. So a better understanding of brain-gut microbiota axis interaction should bring a new insight, and modifying the microbiota composition in the gut would enhance the integrity, as one of the young speakers had shown in an animated slide in the morning. So studies on a new therapeutic approach based on modification of gut microbiota with probiotics, prebiotics, or even fecal microbiota transplantation are awaited in a good study design. And we now have signatures of Parkinson's in the form of microbial signatures, and they are related to the clinical phenotype of developing motor deficits. So they have distinct signatures. As I said, the type of bacterial population, including the strains, are different. So new leads are emerging, and we need to have testable hypotheses on the pathophysiology and then the intervention and the treatment. So microbiota manipulation by supplementing with a known probiotic mix may relieve non-motor symptoms and improve cognitive functions and thereby quality of life. This is just a picture to show eye tracking in the upper panel, and a sensor or a band which we tie, which emits infrared rays and affects the prefrontal cortex, and these are transmitted back, and the gap between the hemoglobin oxygenation and deoxygenation state gives us indirect evidence of brain functional activity. And why we call it functional is we give a task or some kind of stimulus while this is being done. Along with this, to see the background positive control of autonomic stability, we do a wireless physiological monitoring of 16 variables like heart rate, respiratory rates, skin blood flow at 2 sites, skin temperature at 2 sites, as well as the electrodermal activity at 2 sites, and the electromyogram. We can also do a motor planning, which we actually do with just 2 electrodes, and we do the bare shaft or the readiness potentials. Any changes in these pre and post a probiotic intervention can give us a good signature profile of the cognitive state of the subject and improvement by probiotics. And this is also an eye tracking system where we show images. These images could be food images or mood images, and we have designed them ourselves. And we are doing studies in yoga where we have done it, and now we want to take a probiotic intervention and see how attention improves both in children as well as in adults in this spectrum. And our sincere thanks on the left side is our neurodevelopmental team, and on the right side, including Madam Chaudhary, who's sitting here, our team for neurodegenerative disorders. Thank you very much.
Maybe Madam has finished almost 15 minutes before the scheduled time. So can we have 1 or 2 questions, please, quickly?
I'm sorry if I was a bit fast, but lunch hour is approaching, so.
That's okay, we'll take up. Okay, thank you, ma'am.
Sir.
Thank you, ma'am.
And thank you, Probiotic Association of India, Dr. Batish, Dr. Grover, and Dr. Chaudhary, for giving me this opportunity. And it is always good to have a transdisciplinary speakers so that many more ideas can emerge. Thank you.